Ladies and gentlemen, my name is Emem Inyang. I'm a high school senior, and this past summer I had both the honor and privilege of working under the mentorship of Dr. Pierre Alagem with the assistance of Dr. Christopher Don, Ellen Smith in the Laboratory of Molecular Pharmacology, the Center for Cancer Research, the National Cancer Institute at the NIH on a project entitled The Functional Domains of the Replication Origin Binding Protein, RepID. Before I delve into specifics of my project, I would like to provide you some background information to further your understanding of my research. As proposed in 1963, DNA replication initiation involves replicators, which are genetic elements or DNA sequences, and initiators, which are proteins that are sequence-specific that bind to these DNA sequences and recruit other proteins to form a pre-replication complex. Replicators can be located by their ability to initiate DNA replication ectopically, meaning outside of their intended or original location. One of these replicator sites is found on the human beta globin locus, which is located on chromosome 11. Within this locus, there are two regions, RepI, which contained an AT-rich stretch that was essential for its ability to initiate DNA replication, and RepE. Though RepE had an AT-rich stretch, it was a 45 base pair AG-rich stretch was essential for its functioning. Within the AG-rich stretch, there was the AG2 mutant and the AG1 mutant sequence. Now, replicators are futile by themselves. They need proteins to initiate the pre-replication complex. The proteins found in AG2 have been previously discovered to be involved in gene expression, so we set out to find proteins that were bound to the AG1 mutant stretch. It was found that RepID, or the replication initiation determinant protein, also Replication homology domain interacting protein, was sequence-specific to the AG1 site on RepE. This was done via a pull-down assay with a biased and labeled wild-type AG oligonucleotide and mass spectrometry. This protein is essential for the initiation of RepE. It was found that cells' division of RepID had slower cellular replication, reduced replication, initiation events, and affected cell growth. RepID is a fairly large protein. It is 206 kilodaltons, so my lab decided to divide it into 5 different fragments and various combinations of the fragments. Each fragment was tagged with a flag antigen and detected by a flag antibody. This brings me to my methodology. The purpose of my project was to determine which fragments of RepID were integral to RepID's ability to initiate DNA replication based upon cellular localization. We hypothesized that fragments found to be chromatin bound would be essential for RepID's ability to initiate replication. My project had 3 main steps. The first was cell fractionation, where we divided the proteins within the cells based on their location, and immunoblotting followed by microscopy. After plasma transfection, we used human osteosarcoma cells, mainly because they were large and easy to view underneath microscopes, where we expressed the RepID protein. We divided the proteins into cytosolic proteins, nuclear soluble proteins, and chromatin-bound proteins. We quantified them using Lowry protein quantification. We were able to separate the proteins by their size and detect their cellular localization using immunoblotting. Our primary antibody was anti-flag, and our controls were glyceraldehyde 3-phosphate dehydrogenase because it is known to be found within the cytosol. Origin recognition complex subunit 2, or ORC2, because it's known to be within the nucleus, and histone 3, or H3, because it's known to be chromatin bound. After this, we used image J quantification to detect the density of our western band blots. This was confirmed by using immunofluorescence for confocal microscopy. We stained the DNA red using propydium iodide, and we stained our fragments with Alexa-488, which detects green within our pictures. This brings me to our results. Via image J quantification, we were able to detect where proteins lie within the cell. For our full-length protein as our positive control, it was chromatin bound. Fragment 4+5 was mainly chromatin bound, but also nuclear soluble. Fragment 5 was predominantly chromatin bound. Fragments 2 to 5 was chromatin bound. Fragments 3 to 5 were chromatin bound. Fragments 1 to 3 were primarily nuclear soluble. Fragments 1+2 were primarily nuclear soluble. Fragment delta 4, meaning without fragment 4, was both chromatin bound and nuclear soluble. Fragment delta 3 was primarily chromatin bound, and fragment 4 by itself was primarily cytosolic. For our full-length protein, as expected, it is nuclear. Both our empty vector and our wild type being our controls. there is no rep ID expressed. Fragment 4 to 5 is nuclear. Fragment 5 is also nuclear. Fragment 2 to 5 is nuclear. Fragments 3 to 5 were nuclear. Fragments 1 to 3 were cytosolic. Fragments 1 and 2 were cytosolic as well. Fragments delta 3 was nuclear. Fragment delta 4 were cytosolic. And fragment 4 by itself was cytosolic. Let's bring these fun discussion. This is a summary table of rep ID's fragments, localization of using our two methods. The black indicates the majority of localization, and the light blue indicates some localization within the cellular component. We could conclude that with strong concordance between our microscopy and our biochemical fractionation, just meaning that both our results matched up for both methods. Fragment 5 contains a region that acts as a nuclear localization signal for fragment 4, but in context of the entire protein, fragment 5 is not essential for nuclear localization, but it contains a signal that recruits the protein to chromatin. In the future, we'd like to create a fragment 1 to 4 or delete 5 vector just to confirm that fragment 5 is the main fragment that recruits the rest of the protein to the chromatin, co-staining a rep ID with a replication marker, and determining the binding sites of chromatin-bound fragments on chromatin using CHIP, or chromatin immunoblotting. These are the list of people I would like to acknowledge. Thank you. Are there any questions? Yes.
So I was wondering why there weren't many cells expressing the rep ID in microscopy photos?
Immunofluorescence photos. Because we've been using these cells for a very long time, after several passages, some of the cells start to lose expression. That's why they might not be as prevalent in some of the immunofluorescence photos.